Okay, so right now we're going to talk about cell division. You guys have probably already learned about something called mitosis. Well, today we're going to talk about something called meiosis. And you might hear me say the word meiosis while we're talking about this, and that's just something I used to refer to it as just to help me differentiate between mitosis and meiosis because they sound so similar. But just a heads up on that. Mitosis is when we talk about the division of body cells, or you've often heard them called somatic cells. And we use mitosis to replace our body cells. Okay, so like liver cells, spleen, skin, we're going to use mitosis to replace these cells. There's something called meiosis, or some people call it meiosis. This occurs in our sex cells, or what we like to call sperm and egg cells. This is how we're able to reproduce. Um, so there's a difference between mitosis and meiosis. Mitosis occurs in our body cells, our somatic cells. Meiosis occurs in our sex cells, our sperm or egg cells, okay? So meiosis occurs in sexual reproduction when a diploid germ cell produces 4 haploid daughter cells that can mature to become gametes, sperm or egg. Okay, now just real quick before we move on, you should have learned before you got to this point that diploid means that we have 2 sets of genetic information, and haploid means that we have 1 set of genetic information. Now, diploid, we're going to produce in mitosis, diploid individuals are going to produce diploid individuals. So the exact same as themselves, okay? But in meiosis, we're going to take a diploid individual and we're going to produce haploid individuals. So we're going to take the chromosome number and we're going to cut it in half. And the reason why we're going to do this is because sperm is going to meet egg, okay? Sperm's going to meet egg, they're going to join together, and they're going to form a diploid individual. Okay, so just what I said before is our goal in meiosis is to reduce the genetic material by half. And the reason why we do this is because mom's going to give half and dad's going to give half, and we're going to get a diploid individual that's going to be the child. Now, if we took 2 sets of information from mom, 2 sets of information from dad, and we merge those together, we'd get too much information for the child. If we took half information from mom, half information from dad, we join them together, we get just the right amount of information. When we have this going on, when we have like 4 sets or even 3 sets of the genetic information, that's when you start to see genetic diseases like trisomy 21, which is Down syndrome, is caused by getting an extra chromosome or getting an extra set of information. So, again, meiosis is a cell division in 2 parts. It occurs in 2 parts. We have meiosis 1, and then we have meiosis 2. Okay, so the result is 1 copy of each chromosome in a gamete. Okay, so what happens is we go through the cell cycle just like we do in mitosis. We go through interphase, which consists of G1, S, and G2. So G1, we're growing. S phase, we get there and we're duplicating the genetic information. Then we get to G2, and G2 we check ourselves out for cell division. Okay, in S phase we duplicate the information. So what happens in meiosis and mitosis and meiosis 1 is we create 4 sets of genetic information. Okay, that N is denotes our genetic information. So we get 4 sets of genetic information. Well, what happens is we go through a division and we half that information into 2 in, okay? In mitosis, this is where we stop, all right? Because we've taken a diploid individual and we've created 2 diploid individuals. Well, then we get down here, when we go through meiosis, we go through another division, okay? When we go through the next division, we cut it in half again. And when we cut it in half again, we get haploid individuals. That's a really difficult thing for some students to understand sometimes, so you might need a minute with that. So the result is 1 copy of each chromosome in the gamete. Okay, so here we have prophase 1, and we have 4 sets of genetic information. We go to metaphase 1, anaphase 1, telophase 1, and this actually should say diploid individual because we've halved it. Okay, so in early prophase, we have something called tetrads pair. Okay, so we have our tetrads pair, or sometimes you'll hear these called homologs, and that's just where we have two sets of homologous chromosomes, or I'm sorry, 2 sister chromatids. We have our homologous chromosomes pair. This is like, we'll say green represents the chromosome that your mom gave you, and blue represents the one that your dad gave you, okay? These chromosomes have duplicated themselves, and you have 2 sister chromatids that are together, and they're of the same type. So this would be like chromosome 1, okay? So something else also occurs in prophase 1, and this is called crossing over, okay? And this is super important. Crossing over allows... Okay, so during metaphase, these tetrads align along the equator of the cell. In anaphase, I remember ana apart, the tetrads separate and move to opposite poles. Sister chromatids remain attached at their centromeres. During telophase, the nuclear envelope reassembles, spindle fibers disappear, and cytokinesis divides cell into 2. So meiosis 2, the sister chromatids carry identical information. In meiosis 2, they're going to actually separate with 1 copy of each chromosome. With 1 copy of each chromosome, and thus 1 copy of each gene. So I'll show you how this occurs. During prophase 2, we have the diploid is going to actually divide, and we're going to come up with 4 haploid daughters that are not identical to each other. They're genetically different. Every single one is going to be genetically different, because during prophase 1 of meiosis, we had crossing over occur where they swapped genetic information, and when they swapped that genetic information, they made each one of those chromosomes different from each other, and so we're going to end up with genetically different individuals, or genetically different chromosomes in each one of these daughter cells. So prophase 2 happens, and the nuclear envelope fragments, spindle fibers start to form, and centrioles are going to migrate to opposite ends of the pole. Metaphase 2, sister chromatids align along the equator of the cell. Anaphase 2, sister chromatids separate and move to opposite poles. Telophase 2, the nuclear envelope reassembles, the chromosomes unravel, spindles disappear, and cytokinesis divides cell into 2. The results of meiosis is 4 haploid cells, so 4 cells with 1 set of genetic information, and they're all genetically different. So if you look, they're going to be all genetically different. So what happens is during fertilization, we take a haploid sperm cell and a haploid egg cell and we form a diploid zygote, and that's just a fertilized egg. So we take mom's information, dad's information, and we combine them to form this diploid individual. Meiosis allows the creation of unique individuals through sexual reproduction. That's why you and your sister probably don't look anything alike unless you're identical twins, or you have, some similarities, but you're not identical to each other. Okay, so during mitosis, we actually we go through one division, but meiosis goes through two divisions. Okay, the number of daughter cells that we produce during mitosis is going to be 2, where in meiosis it's going to be 4. In mitosis, are they genetically identical? Yes, we're going to produce genetically identical as the parent cell, where in meiosis, no, you produce genetically different cells. The chromosome number in mitosis is going to be the same as the parent cell, where in meiosis it's going to be half of the parent cell. In mitosis, this occurs in the somatic cells or our body cells. In meiosis, it's going to occur in the sex cells. And when does this occur in mitosis? It's throughout life. In meiosis, it's at sexual maturity. Mitosis occurs for growth and repair. So if an individual is growing, you're going to see mitosis occurring a lot in this individual. That's how we start from, that zygote, that fertilized egg, and we get, a baby, and then a toddler, and then a child, and then grows into an adult. This is mitosis that allows that to happen. For meiosis this occurs for sexual reproduction. Okay, so how we get that zygote, those egg and those sperm cells, and then when they join, mitosis takes over. Mitosis occurs in the somatic body cells. Meiosis only occurs in gonads, so our sex organs, ovary and testes. Mitosis produces cells for repair, maintenance, growth, and asexual reproduction. Meiosis, we're going to have only produces gametes, so sex cells, egg, and sperm. In mitosis, the results in an identical diploid daughter cell. And meiosis reduction division results in haploid cells. So, just to kind of talk just a little bit more about genetics, to talk about crossing over and how we get the genetic diversity. Recombination provides for genetic diversity, and the recombination is just what we talk about when we talk about crossing over. We're recombining the genetic information. So the physical exchange of chromosomal material between sister chromatids of homologous chromosomes, and so what that is we have our 2 sister chromatids here. These are homologous. Okay, so this occurs in, so all this is I was explaining. These are homologous chromosomes, which means that they're the same chromosome. That doesn't mean that they're identical to each other. You get one set of this information from your mom, one set of this information from your dad. And we call them homologous because they're the same type of chromosome. So these are probably like chromosome 1 or chromosome 2. They're the same type of chromosome. So like I was saying, you get one of these from mom. Let's say the yellow you got from mom, and you got one from dad. Well, when they go through S phase, and another really bad, just FYI, habit I have is to pronounce homologous chromosomes as homologous. So just FYI, they're pronounced homologous. But anyway, a homologous chromosome or homologous chromosomes are chromosomes of the same type. They're not they're not identical to each other. As you can see, you have A here, little a here. Those denote different genes carried on these chromosomes, or different types of, or different genes that are the same type of gene. So, for example, this chromosome could carry genes for eye color, and so this would carry genes for eye color. Now, the type of, or the specific gene that they carry, they could carry genes on this for blue eye color, while they carry brown eye color on this one, if that makes sense. And just FYI, this goes through S phase, so it duplicates itself, and this is where we have the sister chromatids come into play. So these are sister chromatids, and they're a pair of homologous chromosomes. Okay, so recombination occurs in prophase of meiosis 1. Genetic diversity, this is where this comes from. The letters denote genes. The case denotes alleles, and alleles are just different forms of genes, and we'll talk about that more when we talk about genetics. Wham bam, we have them they cross, and looky there, we have different them on different sides, or different chromosomes. So just so here's another illustration denoting the same thing. We have a duplicated maternal chromosome, a duplicated paternal chromosome, so one from mom, one from dad. This is a sister chromatid. They crossing over occurs in the tetrad. We have genetic variability, so we have part of this chromosome now located on a part of the paternal chromosome located on the maternal chromosome, and vice versa, and this allows for genetic diversity. Okay, and that's it.